Director of the Movement Disorder Center of Arizona in Scottsdale. Dr. Evidente is a fellow of the American Academy of Neurology and a member of the Movement Disorder Society. He has been a frequent presenter and an invited lecturer at national and international conferences. Dr. Evidente is going to speak on the cure for Parkinson's and updates on surgical options.
Okay, thanks, Melissa. So again, my name is Dr. Evidente, and I'm a movement disorders neurologist. And first, let me try to see whether I can share my screen with you. Okay. Okay. And of course, you know, whenever I see patients with Parkinson's, regardless of what the stage is, early Parkinson's, middle stage Parkinson's, or more advanced Parkinson's, almost always patients ask me about cure and whether the medicines that we prescribe them offer cure or not. And the painful answer is always no. The current medicines that we prescribe for Parkinson's are all symptomatic treatment of their symptoms. And none of this have actually offered cure. And previously in our webinar in June, we talked about one avenue that is being explored for possible cure for Parkinson's, which is removing from the brain and the tissues in the rest of the body this abnormal misfolded protein called alpha-synuclein, which forms the Lewy body. And cells that are ridden with misfolded alpha-synuclein tend to degenerate and die, and therefore we talk about the different studies that have been done and are ongoing on how to remove alpha-synuclein or how to reduce the clumping or aggregation of alpha-synuclein in the brain and the rest of the tissues. But today I'm going to talk about a different topic, which is regeneration of brain cells. And of course, we know that Parkinson's disease is a neurodegenerative disorder, and that a part of the brain called the substantia nigra, which produces dopamine, degenerates for a number of reasons, the most common of which would be genetic predisposition plus exposure to environmental toxins. So if we were to treat Parkinson's, we would want to regenerate those brain cells in the substantia nigra. And where exactly is the substantia nigra? It is in a part of the brainstem called the midbrain. And the midbrain is also referred to as the mesencephalon. So mesos is Greek for middle, and cephalon is Greek for brain. So midbrain is where the substantia nigra is. So in this particular figure, I'm just going to orient you guys. So these are the eyeballs over here. Your nose will be here. The back of your head will be here. These are your ears. These are the temporal lobes over here. And these are the occipital lobes. And this is the part of the brainstem called the substantia nigra. I mean, the midbrain rather. And the part of the midbrain that is dark in color is called the substantia nigra. And then the part of the midbrain that is closer to the eyeballs is the ventral part. Whereas the part of the midbrain that is closer to the back of the head is the dorsal part. So the area of interest is the ventral midbrain. And so in this diagram, you will see how the substantia nigra cells or neurons project upwards towards the basal ganglia or the corpus striatum, which consists of the caudate nucleus and the putamen, as well as the globus pallidus. So these projections are important because when the substantia nigra cells or neurons degenerate, this has a trickle-down effect. And so in effect, what happens is that not only are the cells degenerating and dying here, but the cells that are being fed downstream in the striatum, meaning the caudate and putamen, also degenerate. And so most of the strategies that are geared towards regenerating brain cells are geared towards making the corpus striatum regenerate in some shape or form. So some of the earlier attempts use fetal brain cells as donor cells. And it is not just any age of the fetal brain cells that would be viable. In fact, there's a very narrow window. Some believe that the 9 to 11 week old aborted fetuses are the best. And if you harvest their ventral midbrain, and then you transplant this into the brains of either animal models or humans, then the hope is that this fetal brain cells will make connections with the host brain cells, and eventually the transplanted cells would produce dopamine. So this then led to 2 clinical trials, which were done in the US and in different countries in Europe. And this actually attracted a lot of interest in patients, since this was talking about cure now, not just symptomatic therapy. So the initial open label study results were that there was marked improvement of the Parkinson's symptoms. And then if you use a imaging modality called the fluorodopa PET scan, which is the older version of what many of you know nowadays as the dopamine scan or DAT scan. So previously we didn't have that, but what we had and we still have is the fluorodopa PET scan. So this more or less measures the intactness of the presynaptic dopamine pathway, and this will indicate whether the graft cells have survived and whether there's increased dopamine production. And eventually, this initial open label study led to 2 double-blind placebo trials, which were done here in the US, the Colorado Columbus trial and the Tampa study. So for the first study, they noted improvement, particularly in patients who were younger, 60 years or less, and that there was an average of 34% improvement of their motor UPDRS. So the UPDRS is a scale that movement specialists and neurologists like myself routinely administer to our patients. It is a way for us to note whether their Parkinson's is getting better or worse. So the higher the score, the worse; the lower the score, the better. And then in the Tampa study, they transplanted now anywhere from 1 to 4 donors per side from embryos that were aged 6 to 9 weeks. And the results were that the patients that had less advanced Parkinson's disease and received at least 4 donor embryo cells per side improved the most, whereas those that received placebo deteriorated. And then, of course, although many of these patients improved initially, the brain question was the effect enduring? Would the graft cells survive with time? And the answer is yes. So when they did autopsies on these patients that eventually passed on and looked for graft survival, they did find that the grafts did survive more than 10 years in these patients. And then if you do functional imaging, again, do fluorodopa PET scans on these patients, and you do it serially, you will see that along the way, as time goes along, that the grafted cells do survive and continue producing dopamine. And then clinically, how did the patients do? Well, many of these patients improved. Some of these patients stopped taking their Parkinson's medications. But the downside is that a good percentage of these patients develop graft-induced dyskinesia. So in other words, the graft cells eventually produce too much dopamine, and therefore this excessive dopamine causes dyskinesias. And this was an issue because there was absolutely no way to take away, the cells that you've implanted into the brain, and the only way to deal with this was to give them medicines to suppress the dyskinesia. Or some of these patients eventually underwent deep brain stimulation to reduce their dyskinesias. And then some patients lost effect after a few years, and the question is this a result of progression of the disease, or is this as a result of the embryonic grafted cells just dying, and therefore there's less and less of these embryonic cells producing a dopamine with time. And then what was even more alarming was that a few of these patients developed some brain tumors, although they're very uncommon. And then there was this very interesting finding that they found on autopsy in some of these patients, which is the transplanted grafted cells, about 1 to 5% of them develop Lewy bodies. And so the Lewy bodies, which is misfolded alpha-synuclein, were obviously in the host brain cells, and the hope was that the transplanted fetal cells would remain unaffected all throughout. And what they found was that some of these brain cells, actually fetal brain cells, actually were infected, quote unquote, with Lewy body. So here's a picture of the typical Lewy body. This is a brain cell or neuron. This is the nucleus. And then in the cytoplasm, you see this very nice purplish-looking round body, which is called the Lewy body. It's got a halo, which is comprised of what's called Lewy neurites, and these are the filaments which so far have not clumped together and formed the Lewy body. And so these are pathology slides of two patients that made it to autopsy after they passed on, after having received fetal brain cell transplants many years before. And what you see here in one of these specimens is the Lewy body right here. And then in the second patient, you also see the Lewy body right here. So and I guess this would be a function of time, in the sense that as time goes along, many more and more of this grafted cells can get infected by the host brain cells and develop misfolded alpha-synuclein and eventually Lewy body, and eventually degenerate and die themselves. So in other words, they most likely will not last forever. And then the other issue with fetal cell transplants in Parkinson's is that you need a lot of aborted embryos. So if you need 4 or 5 embryos per side of the brain, and not only that the optimal time window would be 9 to 11 weeks, and therefore you will run out of supply very quickly. And furthermore, if you were to harvest the embryonic tissue and store it at 4 degrees Celsius, this is only good for 1 week. And therefore, the time window for harvesting and finding a matched donor would be kind of tricky because of that 1 week period. And then, furthermore, it's difficult enough to find a match when it's one donor and one recipient. When you do what's called HLA typing, and we do this whenever we do organ transplants, it's kind of akin or similar to blood typing. You cannot just receive anybody's blood when you need blood. Your blood obviously has to be matched to the donor. Well, same thing with tissue donation. There is a form of testing for compatibility called HLA typing. And so if you have 4 to 10 embryos as sources for your fetal cell transplants, it will be a nightmare doing HLA typing on 10 embryos at the right time, which is 9 to 11 weeks, and then finding the donor, the recipient for all those embryos, and do everything in a timely fashion. So it's really tricky logistically to achieve all that. Okay, so what about another source, which is more abundant, which are called stem cells? So stem cells have 2 basic properties. One is multipotency, which means they can differentiate into different types of cells. The other term for this is pluripotent. And then they must be self-renewing. In other words, they must be able to divide, to perpetuate themselves, to proliferate. And then there's 2 types of stem cells. The so-called somatic stem cells, which are fixed. They're organ-specific. If you have skin stem cells, they're somatic stem cells. They become only skin cells. If you're talking about neural stem cells or brain stem cells, they only become brain cells. Whereas the so-called pluripotent stem cells can differentiate into any type of cell in the body. So if one were to choose between somatic stem cells, which are fixed in their potency and differentiation, versus pluripotent, obviously the choice would be to use pluripotent stem cells. And then this is something that not many people are aware of, and that is the brain also has stem cells, and they're called neural stem cells. They're not very abundant in the adult brain. They're more abundant, no doubt, in the developing brain. And given that the supply is limited in the adult brain, obviously the best source for neural stem cells will still be embryos. And then if you were to look at the anatomy as to where stem cells are in the brain, and if again were to orient you, imagine your ears to be here on this side, the top of your head over here, the bottom of your head over here. And this is the so-called lateral ventricle. So outside of the wall of the lateral ventricle, the so-called subventricular zone, which is a rich area for stem cells. And then if these are the temporal lobes, the medial part is right around here. There's a structure called the hippocampus, and the hippocampal dentate gyrus is another rich source. And then the olfactory bulb, which has to do with olfaction or sense of smell, is somewhere here in the bottom. And then the striatum, which I alluded to earlier on, which is the caudate and the putamen, is a less fertile source for stem cells, even in embryos. Okay, so pluripotent stem cells sources are either embryos or you induce them. So when it comes to embryos, the sources will be either spontaneous abortions, aborted fetuses, or this is also available in clinics that do in vitro fertilization. So these so-called unused embryos can be repurposed and used as a source for embryonic stem cells for cell transplants. And then what's even more exciting are the so-called induced pluripotent stem cells. So the way this is done is you get a somatic cell, which is supposed to be fixed in terms of its fate. You reprogram it genetically, and then you make it into a different kind of cell. And so what are the most common somatic cells which are used for to produce induced pluripotent stem cells? So the richest sources are the skin and the blood. And then there's other sources like fat, cartilage, bone marrow, and also the umbilical cord of blood. Now the pluripotent stem cells can come from one cell, but that's called autologous, which is the preferred source, meaning because there's less chances of rejection, versus another person that's called allogenic. And if you have allogenic sources of your stem cells, you obviously need HLA typing. And then it undergoes this really complex, difficult to comprehend and more so difficult to explain process of inducing first with some genetic manipulation, and then exposing those pluripotent stem cells to cytokines. It really depends on what cytokines you use. It is some sort of a formula or a recipe, and the recipe for producing dopamine cells out of this pluripotent stem cells consists of this sonic hedgehog, the fibroblast growth factor A, and the glycogen synthase kinase 3 beta inhibitor. So in other words, if you use this methodology, the pluripotent stem cells that you get from the skin or from other source, the blood, eventually will become dopamine-producing cells. Okay, of course, any of these therapies will have some risks. One would be infection. And so one way to avoid that is obviously making sure that if you go for iPS cell-based therapy, that you go to a reputable center, and a center, a lab with good, that adheres to good manufacturer practices in accordance with FDA regulations and other regulatory agencies. And then there's a small chance of tumor production. In fact, this has been seen not uncommonly in years after, not only in Parkinson's, but in other diseases that have been amenable to stem cell treatment. For instance, here in the side of the pons, which is the part of middle part of the brainstem, you see this tumor over here. which is not supposed to be there. And then here in the lower back, in the lumbosacral region, these are the vertebrae, these are the discs, this is the back of your lower back. And then right here, it's supposedly the nerve roots. And you see this thing here, which is a tumor, which is not supposed to be there. So both this tumor in the spinal cord and this tumor at the side of the brainstem developed in a patient who had received neural stem cell transplantation. And then similar to the embryonic grafts, there is a chance of dyskinesias. And the risk factors for this appear to be, looking back at those patients, it appears that those that already have levodopa-induced dyskinesias to start with, and those with severe motor fluctuations simply be more at risk for graft-induced dyskinesias. And again, this could be very difficult to manage because there's absolutely no way you can take out those cells that are producing too much dopamine. And then what about umbilical cord blood stem cells? So they've actually done studies in rats, and you can actually induce Parkinson's in rats by exposing them to certain toxins, like for instance, MPTP in monkeys. They also use this substance or toxin called 6-OHTA in mice. And then if you inject this directly into the striatum, it will produce an animal model of Parkinson's. So the mice will develop symptoms of Parkinson's. They'll be slower, and they may have tremors around their whiskers. So if you were to harvest, therefore, the umbilical cord blood, and this is usually rich in different types of stem cells, including neural stem cells, and then now you transplant those into the striatum, what they found out was that this umbilical cord blood stem cell differentiated into dopamine neurons within 4 months, which is really promising. And there was also improvement of the motor deficits in the rat PD model. Now, I'm not aware of any studies yet ongoing in any part of the world on umbilical cord stem cells for actual Parkinson's patients. Many patients have asked me in the past about the cost effectiveness or the wisdom of storing umbilical cord blood in babies that are born in their families. And surprisingly, it's actually not that expensive. And from my research, the actual harvesting of the cord blood in the first year of storage is going to cost somewhere around $1,000 to $2,000, and the yearly cost of storage is somewhere in the order of about $150 to $200. So it's not a big expense. Although obviously, given the lack of studies as to whether that would be useful for Parkinson's, is obviously a gamble in one's part, and you kind of have to just place your bets that this will be useful in the future. If not for Parkinson's, perhaps for something else, either for yourself or for other individuals who are of the same HLA type. And then what about gene therapy? So the basic principle with gene therapy is to replace bad DNA with good DNA. And so the principle would be to introduce genes, which are therapeutic genes, to either replace a bad gene, to silence a gene that is bad, or to correct a gene that's faulty. And the way to make this work is you first need a viral vector to infect the brain cells. And obviously, it cannot just be any virus. It has to be a virus that is innocuous to humans. It must not be pathological to humans. Secondly, it must be infectious in the sense that it will infect brain cells quite effectively. And thirdly, it should not induce a very strong immunological response. Obviously, you're not going to be interested in the virus like the COVID virus, which is not only highly contagious and infectious, but also induces a very strong immunological response. You don't want that kind of virus. Now, the one that fits the tab is the Adeno-associated virus, or AAV for short. So we have the viral vector. You insert the gene into the viral vector, and then you introduce the viral vector and the gene into the brain of the host, and the hope is that the gene gets incorporated into the host DNA, and then now you monitor the clinical response In this case, the motor scores and the symptoms of Parkinson's, and you also do PET scan, fluid PET scans on these patients looking for evidence of increased dopamine production. For instance, as far as the PET scans are concerned, this is somebody who had a PET scan before transplantation into the striatum. What you see in red here would be the hot spots for dopamine. And then 6 months after the transplantation, you see more red areas in the striatum, which tells us that there's now more dopamine producing cells. So this is one way for us to determine whether the grafts are, number one, surviving, and number two, are they producing dopamine, aside from the fact that we monitor these patients clinically. And then as far as the different kinds of gene therapies, some are non-disease modifying, whereas the others are disease modifying. So non-disease modifying means you just treat the symptoms. Whereas disease modifying means you slow down the progression of the disorder, you stop its progression, or even better, you regenerate the brain cells that have been lost or that have died. And one of the more commonly publicized studies on the non-disease modifying gene therapy involves a gene that is responsible for the production of an enzyme called AADC, or aromatic amino acid decarboxylase, or dopa decarboxylase for Parkinson's. There's evidence that this enzyme is reduced. And if I were to just give you a little bit of biochemistry, this is L-dopa, which is cinnamate. You ingest it, it crosses the blood-brain barrier, and then inside the brain, using an enzyme called dopa decarboxylase, levodopa or L-dopa gets converted to dopamine, and this is the one that stimulates the receptors, the dopamine receptors in the brain. And so an early phase 1 study in 2009, which studied about 10 Parkinson's patients in the moderate stage, they used this approach of the AADC gene, and they used an AAV-2 viral vector. This was implanted into the striatum in both sides. And what they found was increased on time without dyskinesias, which is good, which is the best on time, and then reduce off time at 6 months. And then there was improvement of the motor scores, the UPDRS, by 30%, both in the off state and the on state. And then if you do fluorodopa PET scans on this patient, there was definitely evidence of increased dopamine production by about 30 to 75% at 6 months. So really not bad. So this therefore led to a phase 2 trial, which is actually ongoing, which is sponsored by Neurocrine in collaboration with Voyager. And this was a gene therapy for AADC, which is implanted into bilateral putamen. But as with most studies that eventually make it to approval by the FDA, SHAM surgery has to be performed. And the way that's done is some patients will have a burr hole drilled in their skull, and they will think that they had the surgery when in fact, no cells were implanted into their brain. And now they compare patients who had the burr hole plus the gene implanted into their striatum versus those that only have the SHAM burr holes. And believe it or not, patients that had only the SHAM surgery also had a positive effect, and this is the placebo effect, which is pretty strong in Parkinson's trials, and it's not something to laugh at or minimize. And that is precisely why it is very important in development of Parkinson's therapy that the treatment arm should be no doubt superior to the placebo before it can get approved. So Restore 1 study is a phase 2 trial. They want patients ages 40 to 75, 12-month follow-up, 21 visits, AADC gene versus SHAM. They need about 85 patients, and this is ongoing. And then this will be followed by a phase 3 trial, which I believe is delayed because of the pandemic, called Restore 2. I think this will start hopefully towards the end of 2020 and maybe the beginning of next year. Limitations of this approach, since AADC is an enzyme that converts L-dopa to dopamine, you still need levodopa. In other words, patients will still need their medications. It is just that the L-dopa with this approach will be more effectively converted to dopamine in their brains. And then secondly, the AAV vector cannot carry large amounts of genes or multiple genes because it's a relatively small vector. Therefore, that led to finding of a bigger viral vector, and that is where the lentiviruses come in, because the lentiviruses can carry multiple genes. And then if we're talking about Parkinson's, we talk about one gene which is important for the production of dopamine, but there's other genes that we can consider in gene therapy for Parkinson's. So for instance, just to orient you again, this is the presynaptic terminal, this is the synapse, this is the postsynaptic neuron over here. These are the dopamine receptors, these are the dopamine molecules. And then L-dopa becomes dopamine through AADC, and the gene that regulates it is called the AADC gene. But the precursor to L-dopa is tyrosine. And then there is this enzyme called tyrosine hydroxylase. And there's a gene that regulates that called the tyrosine hydroxylase gene. And then you need a cofactor called BH4, tetrahydrobiopterin. And then this particular cofactor comes from GTP, and you need another enzyme called GCH1, which is regulated by a gene called GCH1 gene. So can we put all three genes which are involved in the production of dopamine into one virus? And the answer is yes. It has to be a large lentivirus, and the first one developed that can carry all three genes called Pro7. And this was developed by Oxford Biomedica. In 2014, they initiated their phase 1 and phase 2 studies. This was an open label trial. 50 patients were done, 12-month follow-up, and there were only 2 sites in France and in the UK. And there was bilateral injection of this Pro7 into the putamen. And what they found was significant improvement of their motor scores at 6 and 12 months. And not surprising, again, the most common side effect was increased dyskinesias. And then they went one step further. They developed something even better than Pro7 called AXO-Lenti-PD. And this was done first in monkey studies that were induced to have Parkinson's through a toxin called MPTP. And then this eventually led to a phase 1 and 2 study in England and France called the Sunrise PD study in 2019. That is when this was started. And the initial data showed that this was very well tolerated. So far, they had released data on 2 patients at 6 months follow-up with improved motor function. We will hopefully know more about this study by next year or the year after. And then what about disease-modifying gene therapy in Parkinson's? And what comes to mind is a gene called the GAD gene, or glutamic acid decarboxylase. So GAD gene is involved in the production of a neurotransmitter called GABA, which is inhibitory. Now, is there evidence of excessive excitatory tone in certain nuclei in the brain in Parkinson's? The answer is yes. There is evidence of hyperactivity in the subthalamic nucleus, which uses a substance called glutamate, which is excitatory. So just to orient you again, so you have the thalamus over here. So subthalamic nucleus or subthalamus is right here, which is above the substantia nigra, which is around here. And then this projects, as I said, the substantia projects to the striatum, which is decoded over here in the putamen. And in fact, the subthalamic nucleus is the most common target for deep brain stimulation. So many of you who've had DBS or know somebody who've had DBS would know this target very well. And then just to review a little bit, you know, how the basal ganglia works and how it incorporates the different structures in the brain. Here's the striatum. Here's the dopamine from the substantia nigra, which influences striatum, and the subthalamic nucleus is somewhere here in that circuit. And then what happens in Parkinson's is that this particular part of the circuit called the indirect pathway is overactive. And so glutamate is the neurotransmitter in this particular nucleus. So one way to inhibit this is to introduce GABA into the STN and inhibit it so that the output from the STN will be reduced, the hyperactivity. So this led to a phase 1 study in 2007, the unilateral injection of the AAV-GAD gene vector molecule into the STN in MPTP monkeys, and they found decreased bradykinesia and tremor. And this eventually also, a few years later, led to a phase 2 study sponsored by Neurologics, AAV-GAD versus SHAM in 37 PD patients. And the results were actually very promising. It's a shame, though, that because of lack of funds, there was no follow-up study to this. And then what other disease-modifying genes can we use using this technology? Well, we can use neurotrophic factors. So neurotrophic factors are substances or factors that increase survival of brain cells. And the most common ones that we've heard of are GDNF, BDNF, nurturin, artemin, persiffin, and VGFF. So in animal studies, these are well tolerated. There was increase in striatal cells and improvement of Parkinsonism in the animal models. Unfortunately, one of these studies, which is a double-blind sham survey-controlled study of AAV2 using nurturin in the putamen or the putamen plus the substantia nigra, revealed discouraging results. However, earlier this year, an open-label study was initiated by Brain Neurotherapy. This is a phase 1b study of GDNF. It's a different neurotrophic factor this time, which seems to be more promising than nurturin. It is actively recruiting, and the closest to Arizona are geographically are UCSF and Ohio State University for those that might be interested in contacting them. And the study will infuse the GDNF AAV2 bilaterally into the putamen. So this is an actively recruiting study. And then what about gene therapy to convert non-neurons into dopamine-producing neurons? So in other words, you have the neurons, which are the yellow things here, or structures, which are the main players in the brain. And then you have the astrocytes, which are green, which are support cells and the nourishing cells in the brain. And then you have other non-neurons in the brain, including the microglia, which are involved in the defense against foreign substances and microorganisms. And then you have the oligodendrocytes, which produce myelin, which envelops the axons of the neurons and therefore facilitate the transmission of nerve signals. So is there a way to convert non-neurons into dopamine-producing neurons? And the answer is yes. An animal study was published in Nature, which is a very highly respected basic science journal in June of this year. A study out of UC San Diego, a single treatment of PTB antisense oligonucleotide was injected into mice midbrain, and this inhibited a gene called PTB. And what is the result? That resulted in conversion of the astrocytes, which I showed you in the previous slide, into dopamine-producing cells. Wonderful. So this provided proof of concept that this was possible, this was feasible, and therefore this is something that can be explored in the future, hopefully next in humans. And they also observed that the mice that had this treatment had improvements of their PD symptoms. Okay, so most of the things or interventions we've discussed so far involve surgery, but not everybody is a fan of surgery. In fact, some of my patients are deathly afraid of surgery. The moment I talk about surgery, they kind of zone out and start talking about something else, or just refuse to discuss it. And I respect that very much. So is it possible to do gene therapy without surgery? So in theory, how can one envision this? So one way to envision that this may work is to inject into the vein the same viral vector with the gene incorporated into the viral vector, and then give that viral vector some sort of a mechanism or a GPS to home into the brain cell, into the brain, and not only to any part of the brain, but to the parts of the brain that need to be influenced, which in the case of Parkinson's would be the striatum or the basal ganglia. And so there's two hurdles to this concept. One is there has to be a way for the gene viral vector to cross the so-called blood-brain barrier, which is supposed to protect the brain from bigger molecules and foreign substances. And number two, the viral vector must be able to seek the target. So it must have some sort of a GPS. So we have to find a viral vector that homes into the striatum innately. And so just as a review of the so-called blood-brain barrier, so this is a capillary, which is the smallest of blood vessels. This is the normal blood vessel versus the blood vessel or the capillaries in the brain. So the non-brain capillaries have holes or pores where molecules that are big enough can squeeze through and exit from the lumen of the blood vessel into the tissues. Whereas if you look at the capillaries in the brain, the pores are really, really tight. And they're called tight junctions. They have no pores. And also, not only are the capillaries tight in the so-called junctions, but they're also surrounded by the glial brain cells, and which add a second layer of a barrier from substances to go from the lumen of the blood vessel to the brain. So is there a way to penetrate the blood-brain barrier? So first, let's look at the viral vector. And I'm going to first talk about the AAV vector, since this is the virus of interest that we discussed earlier. So here's the typical AAV virus. So this is its nucleic acid or DNA, and then it's got this covering or shell called the capsid. And then the capsid consists of 60 proteins, which in this cartoon is represented with different colors. So there's 60 different proteins. So can we manipulate the proteins? For instance, insert some amino acids so that the AAV capsid is modified and will be able now to penetrate the blood-brain barrier? And the answer is yes. They've found ways to modify the proteins in the capsid or capsule or the envelope of the AAV, and therefore be able to cross the blood-brain barrier. And there's about 13 strains that they found out, of which the one that penetrates the blood-brain barrier the most is this strain called AAV-PHP.B. And then there's another way to actually increase the permeability of the blood-brain barrier to allow different substances, including medications and so on and so forth, to cross the blood-brain barrier more effectively, and that's called focus ultrasound. And some of you would have heard about focus ultrasound, which is an alternative to deep brain stimulation in patients with severe essential tremor. So it is a surgical-less way of lesioning the part of the brain called the thalamus through ultrasound, which if you focus, hundreds of these beams of ultrasound to one fine target and magnify it, so to speak, into that one target area, then you will create a lesion. So this technology can be used to increase the permeability of the blood-brain barrier. And then the second question is, does the AAV-PHP.B virus have an innate affinity for the striatum? So the one way to test that is to insert a piece of DNA into any of these viruses that you want to test, and then this particular DNA will make it fluoresce. And so if you test different viruses, for instance, the AAV9 virus, and this is the striatum over here, and you see that it doesn't fluoresce very much. So this tells me that the AAV9 virus doesn't have a great affinity for the striatum, which is right around here. And yet, if you look at the AAV-PHP.B virus, you see that it fluoresces in many parts of the brain, and no doubt the fluorescence seems to be even higher in the striatum. They calculate about 92 times higher in the striatum than it is in the rest of the brain. So it's a good vector. It homes into the brain. So the only thing lacking now is to choose the gene or the substance that you want to write on and introduce into the brain. And this can be done in either early Parkinson's disease, or you can even do this in patients with pre-symptomatic genetic Parkinson's. So if there's a patient that is a carrier of a certain gene that predisposes to Parkinson's, this might be a therapy that is amenable to improvement with that technology. And then in advanced Parkinson's, this technology can also be used to reprogram the striatal astrocytes, which are the support cells, they're non-neurons, to become dopamine neurons. And then this particular technology has also been utilized for the GBA1 gene, which is, believe it or not, the most common gene defect that is seen even in sporadic Parkinson's. So the GBA1 gene produces an enzyme called glucocerebrosidase. And those that have the GBA1 gene mutation are 5 times more at risk for Parkinson's, and they're also more at risk for more severe Parkinson's. So this enzyme called glucocerebrosidase, and where do you find them in the cell? You find them in special organelles called lysosomes. And these are the lysosomes right here. And the lysosomes are organelles that are involved in digesting food particles. They're also digesting excessive materials which are not needed. They're also involved in digesting toxins and waste products, so that the toxins and waste products do not accumulate within the cell. And so if there is a defect in the production of these enzymes, including glucocerebrosidase, like for instance, you have the GBA1 gene mutation, you produce less of these digestive enzymes. So therefore, what happens is toxins and waste products accumulate within the cell. And eventually the cells will die. And as we discussed in the previous webinar, there is more misfolded alpha-synuclein in these cells, and therefore these cells die even more easily. So they've done this in mice studies. And what they found was a significant increase in glucocerebrosidase, which is, again, the digestive enzyme, which is good, and then a much lower alpha-synuclein in the brain. And so this now led to the so-called PROPEL study, which is sponsored by a company called Prevail Therapeutics. This is a phase 1/2A study of a product called PR001A, which is the GBA1 gene. But they will first do a gene test on the participants. They have to make sure they have at least a mutation of one of your alleles for the GBA1 gene, that you're at least a heterozygote. And then this gene viral vector is injected into the intracisternal spaces, most likely in the spinal fluid in your back. And then 2 doses will be tested, or should I say, the surgery rather will be done in this case. Not injecting, but surgery will be done. And then they will compare these 2 doses versus sham surgery, and the duration of study is 5 years. And they need 16 participants, and this is active day recruiting in a number of sites. And then lastly, there's this really fancy way of genetic manipulation called gene editing. And then most likely what will be amenable as far as Parkinson's are concerned are those with mutations in some of the genes for Parkinson's. So mind you, currently there are about close to 20 genes for Parkinson's, and the most common of which is GBA1 gene and the LRRK2. And there's this tool called CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, which is being used for different diseases, or even in plant studies or animal studies that is being used to edit genes. And the basic principle is that it involves two molecules, a single-strand RNA called a guide RNA, which is known as CRISPR, which will home into the DNA sequence of interest. And then once you find that DNA sequence of interest, you use a molecular scissors called Cas9, and you edit it or you take it away. So I'm going to play a video now that I found in the internet, in YouTube, which very nicely summarizes what CRISPR is all about.
CRISPR. It's science's potential secret weapon for treating inherited diseases like sickle cell anemia and muscular dystrophy. So, how does CRISPR work? To be specific, CRISPR is a system that guides a protein called Cas9 to cut DNA, basically editing your genes. Let's say you want to fix some faulty DNA in a genome. It's a three-step process. To make the CRISPR system go, you provide the genetic address for where the bad stuff is in the DNA. Cas9 uses that sequence to find the bad stuff. Then it starts cutting. How does this all play out? CRISPR uses a guide molecule of RNA, as many as 105 letters long, 20 of which match the target sequence in a gene to find the bad stuff. Swap out the guide RNA, and Cas9 can be directed to millions of different sites. Think of it like an itsy-bitsy tracking device. Then what? Cutting to delete, or cutting to edit. Delete. Use CRISPR to cut a gene, which can deactivate the bad gene. Edit. CRISPR cuts, but now with a template to make a DNA repair. After the repair, you've got a fresh, clean gene. This seems pretty simple. Is CRISPR really such a big deal? Yes, it most certainly is. Consider sickle cell anemia. This disease causes crippling pain, organ failure, and premature death, all because of a single misspelled letter of DNA. Change that, and poof, goodbye sickle cell anemia. Some scientists believe CRISPR could even be used to treat diseases like muscular dystrophy, and they've already shown this works in mice. Okay, so getting the CRISPR ingredients into a human body isn't going to be easy, but scientists are working on it.
Okay, so I really love that video since it concisely explains what CRISPR is all about and about the principles behind it. Unfortunately, CRISPR can be used in a bad way as well. For instance, you can actually genetically modify babies if you wish with this technology. And the way to do this is to alter the DNA in the reproductive cells. And so you literally can choose, the sex of the baby, the physical features, what diseases they will be protected against, and so forth, and so on and so forth. So this creates a moral and ethical dilemma, and there actually is a ban almost universally on doing this, and this is how a Chinese biophysicist got into trouble in 2018 when he, in an international meeting in Hong Kong, he actually announced that he for the first time in the world that he created the first genetically modified babies with good intentions though, because his intention was to remove the CCR5 gene, which predisposes to HIV. So if you remove that, these babies become resistant to HIV. So this is something very important for couples where one of the couple is HIV positive. So the intentions were good, but again, this is a technology which has moral implications. But let's talk about the good applications for this, especially for what we're interested in, which is Parkinson's. So is this moving forward in the arena of Parkinson's? And the answer is yes. And in fact, the Michael J. Fox funded a research which has to do with downregulation of alpha-synuclein using this approach. So So there is a gene called the SNCA gene, which is the alpha-synuclein gene, which is responsible for production of alpha-synuclein. So the hypothesis is, the working hypothesis is, if you do gene therapy using CRISPR, can you lower the alpha-synuclein production and alpha-synuclein clumping? And so this seems to be a very sound idea, and we will see over the years how this research goes along. So back to my earlier slides is, I think we're coming closer and closer to finding the cure for Parkinson's. Ever since I was a fellow more than 20 years ago, almost every month or so, I read something in the medical journals or read something in the internet or see something on TV about the so-called cure for Parkinson's, and yet so far we have no cure. So whenever you read something that is touting to be the cure for Parkinson's, most likely that's bogus. But as I mentioned in my previous webinar, I think one approach that will work is removing alpha-synuclein from the brain for antibodies to rehash, repurpose medications that will reduce the clumping or aggregation or dissolve what's already in your brain or in your tissues. And now, based on what we discussed today, use stem cell therapy and gene therapy to regenerate regenerative in Parkinson's. And with that, I'm going to end, and I'm going to open the floor for any questions.